Welcome to the channel, Cancer, Metabolic, Not Genetic. I'm your host, Vince Gledhill. In today's episode, a fascinating insight from Professor Thomas Seyfried on how cancer starts in the first place. I give you Professor Thomas Seyfried.
Yeah, so, the definition of cancer, and I may have mentioned this before, is cell division out of control. So what happens to the first or group of cells that become dysregulated in their growth? Because ultimately, this is what the problem is. You have most of the cells in our body are in what we call a differentiated state, a quiescent state. They're all arranged in order in our various organs and tissues, and they all contribute together to the society of cells in the body, which ultimately is us. Whether it's in the brain, the liver, you have liver dysfunction and your brain responds in a way. The brain would have a problem. The liver would respond in a way. All of our systems are integrated through blood networks, through immune systems. So we are actually a very highly tuned, integrated machine. What happens, though, to a cell in, say, a breast or a bladder or a colon or lung, that would make that cell become dysregulated in its growth? So, there's been a lot of ways that cancer has been produced in cells and tissues of our body, and most of it has to do with a chronic disruption of the cellular microenvironment. Through irritation, through a viral infection, through a blocked blood vessel that would cause a chronic or restricted a hypoxic environment. The problem is that and I say chronic because most acute damages to cells and tissues will lead to rapid death. And as Otto Warburg said, you can never get a dysregulated cancer cell from a dead cell. So there has to be some time frame when this regulated cell, this quiescent differentiated cell, has an opportunity to become dysregulated. And what's responsible for the quiescent regulated differentiated state is the function of the energy metabolism within that cell. And that's controlled by the organelle called the mitochondrion. It's kind of a spaghetti network that exists in the cytoplasm of the cell. That network interacts with the nucleus. The nucleus and the mitochondria within a cell have a very intimate communication system from signaling pathways and such like this. But a chronic insult to a cell or a tissue leads to a gradual loss of energy through oxygen. So to prove that, you're breathing, I'm breathing, most living people, most animal, everything gets energy. They breathe oxygen, get oxygen in, either through the gills, through the lungs, or whatever. That oxygen serves as an acceptor, a final electron acceptor for the electron transport chain in the mitochondria that generate large amounts of energy very efficiently. However, if that energy efficiency is chronically interrupted, not to the point where you kill the cell, because if there's no energy, death, you can't be live with no energy. So what happens in the cancer process is that the energy is transferred from oxidative phosphorylation, breathing oxygen, to an ancient pathway of what we call fermentation. The technical term is substrate level phosphorylation, a type of energy mechanism that existed for all living organisms that were on our planet before oxygen came into the atmosphere some 2.5 billion years ago. So these heirlooms of energy metabolism exist in most of the cells of our body, these old ancient heirlooms, but they play a very, very minor role when we're breathing oxygen. They have a very low production of energy. If these OXPHOS, energy through oxidative phosphorylation, is corrupted or deficient or anything like that, the ancient pathways are upregulated. And the mitochondria send a message to the nucleus of the cell, and the nucleus of the cell sends out a messenger that upregulates transporters for glucose and glutamine, the two fuels that are necessary for these ancient pathways to maintain viability. The problem with that is that that same organelle, the mitochondria that's responsible for differentiation and quiescence, also controls the cell cycle and the differentiated state. So when that organelle becomes corrupted and the cell falls back on the ancient fermentation pathways, the controller of the cell cycle is not there. And what happens? The cell starts to divide exactly the way it did 2.5 billion years ago, because all of the cells at that time were dysregulated in their growth. They just grew and grew and grew and grew until the fuels, fermentable fuels, disappeared in the environment and the cells would die. So this whole cancer thing is not a mystery. These cells are simply falling back, these cancer cells. And when you look at them, in every major tissue of every major cancer, you look at the mitochondria in the human cells and you find them defective. You find them few in number, their morphology is abnormal, and their function is abnormal. So if that's all abnormal and structure determines function, then these cells are dividing and dysregulated because the very organelle that's controlling this process is dysfunctional. And these cells then are growing based on the availability of the sugar glucose and the amino acid glutamine in the microenvironment of those cells. And as long as those two fuels are present in sufficient quantities, those cells will be growing in a dysregulated way, and they become worse and worse and worse until you get the spread, which we now know what underlies that. That's another story that would require some level of explanation. But we know basically, I understand basically what causes cancer, how it develops, how it metastasizes, and basically what we need to do to stop it in a non-toxic way.
To subscribe to the channel, click here. And if you haven't watched it, this is a great presentation by the professor over here.